Good morning, good evening, everyone. Let's wait a couple of seconds to allow all the people to reach us. And in the meantime, I will start with a couple of announcements. So remind that the registration and abstract submission is now open for the Cancer Cachexia Society that will be held in Turin in September 2025. Here you have the link to the website of the society. You can register today and you can submit later, and you can find the different deadlines on the website. And moreover, also this year we have the next generation researchers in cancer cachexia, the Communication Award, and also a scientific image context. And you will receive info about this competition very soon. So save the date because in April we will have our popular trainee talks. The Education Committee have finished to review the applications. We received many, and now we have selected 3 speakers, and we will announce the names very soon. And then in May, we will have the seminar with Professor Barry Laird and Christine Hustad. They will present the My Path project, that is the integrating nutritional care into routine cancer treatment. And so now let me to introduce our speaker today, Christian Frezza. Christian, thank you for accepting our invitation. I have to say that it's very hard to introduce Christian because he has an astonishing and successful career. So I will try to do my best in summarizing your achievements. So Christian Frezza is a professor of metabolomics in aging at the University of Cologne, Germany. And he has a long-standing interest in the role of mitochondrial biology and metabolism in human diseases. He got a PhD at the University of Padua, where he made very important discoveries published in Cell on the mechanism of action of OPA1. Then, at the end of his PhD, he moved in the laboratory of Professor Gottlieb at the Cancer Research UK Beeston Institute in Glasgow. And here he published a Nature paper providing new mechanistic insights into the role of dysregulated metabolism in cancer. In 2012, he was recruited to a program leader track at the Medical Research Council Cancer Unit at the University of Cambridge, where he started his lab, continuing to investigate the role of dysregulated mitochondrial metabolism in cancer using a combination of computational and experimental approaches, and also continuing publishing in Nature and winning very important and competitive grants such as the ERC Consolidator Grant. So in 2021, he joined the Cologne Excellence Cluster for Aging and Aging-Associated Diseases at the University of Cologne in Germany, where he is currently working. So today, in his talk, he will give an overview of metabolomics strategies to analyze cell metabolism, and how to properly present data, and how he used metabolomics in his model of renal cancer. So this could be useful not only for the trainees for whom we specifically design and thought this seminar, but also for those who have to review grants or paper showing and containing metabolomics data. So thank you very much for accepting our invitation and for your generosity in preparing this seminar specifically for us and for our trainees. So thank you, Christian, again. I'm very grateful that you accepted our invitation.
Thank you, Roberta. Can you hear me? Okay.
Yes, yeah. Perfectly. Okay. You can share your screen.
Yes. First of all, thanks very much, Roberta, for the intro. You have been too kind. I just wanted to remind people that yes, I haven't published only in in Nature. These are only a few. There are other work that I've seen, but I will not. In other journals, even that I will mention probably, I will not mention the journals, but I will show some of the results. Okay, it's just scientific life is not only made of nature and higher impact papers, right? I just wanted to also thank you for giving me this opportunity. It will be the first time that I give a presentation like this. So I hope you will understand what I want to say. And if you have any question, I think you can even interrupt me. I don't know if this is normal in this type of
People can write in the Q&A
yes.
their questions. Yeah.
Yes. So because again, it's my first attempt. I will do my best, and I hope you will find it interesting. Okay. So without further ado, I will just start by first of all asking. You asked me about metabolomics, and I thought I would start from the very bottom, that is with the definition, because you will be surprised that even nowadays, sometimes we get requests from people collaborating and asking whether we can measure proteins or lipids with our machines, even though they know that we do metabolomics. So I just wanted to clarify what is metabolomics and what does it measure. And let's split the words in two. The first, of course, is metabolomics. Metabol refers to the analysis of small molecule metabolites. And just to maybe even more be precise, these molecule metabolites are different from, for instance, lipids. In that case, we will be talking about lipidomics. And this is technically and also conceptually very different. And omics, of course, is the term that is used nowadays very often. You have seen it and you've heard it. And it's referring to an analysis of all the constituent of a specific layer of complexity that we investigate. So we go from transcriptomics to proteomics to metabolomics and many others. So metabolomics, as a definition, is the comprehensive study of chemical processes involving metabolites that are key components of metabolism. So this, I know, maybe you will find this offensive, but I thought maybe just to explain what is metabolism in a nutshell, just for those of you who are starting. So here, this is a very, very simplified view of the cell and of metabolism. So I will define metabolism as the process through which nutrients and foodstuff is converted into energy and biomass. And as you know, I am a very big fan of mitochondria. Mitochondria take a big role in metabolism, in particular because they use oxygen to oxidize the foodstuff and allow the production of energy. During all this process of conversion of nutrients into energy and biomass, some molecules are produced, are broken down from the complex food, and these are exactly metabolites. So these metabolites are small molecules that can be eventually converted into other small molecules, and eventually either converted into energy or into other molecules into biomass generation. And you might know that nowadays, thanks to the advance of metabolomics, we have a fairly clear understanding of the metabolites that are present in the cell and in the human body. So this shows how nowadays we can deconstruct the human body, and we can come up with its entire metabolome. And this is a picture that you have seen probably 100 times. This is a map of cellular metabolism from kegs. And this includes all the known reactions in the humans, but also in other organisms. And just to give an idea, in the Human Metabolome Database, this is the largest database known so far that contains all the metabolites. There are more than 40,000 metabolites listed there. Now, how do we analyze the metabolites in our cells. And here, again, I just want to be very general, just for giving you an idea. There are two main approaches for analyzing metabolism. One is via nuclear magnetic resonance, or NMR, and I'm not going to talk about this in details today. And the other analytical approach that can be used for analyzing metabolites is called mass spectrometer. And you might have heard much more often about mass spectrometer-based metabolomics. And I want to show you how the pipeline works, because it's very complex. It's not only about getting the mass spectrometer, but actually nowadays to perform the most standard metabolomics analysis, you need two pieces of equipment. The first one is a liquid chromatography setup, and the second one is a mass spectrometer. I will tell you more in detail about these two parts in a second. So how is mass spec pipeline defined? First of all, you have to know that you can start from more or less any type of substrate for analyzing the metabolite content. And you can start, and we do metabolomics on the blood, on any bodily fluid. Nowadays, you can collect tissue, interstitial fluid from the muscle. You can collect saliva. You can collect even tears from the eye. But more often, we do metabolomics from the cell culture and from media of the cell culture, and also from the mouse tissue. And a very important step of the metabolomics pipeline is to extract the metabolites from this substrate. And of course, this is very complex. Depending on the type of extraction that you do, you can extract different types of metabolites. I don't want to go into details, but I'm happy to elaborate. Now, imagine that after taking your tissue, you isolate metabolites. And then imagine these metabolites are indicated here as these dots. These are all the chemicals that I color-coded. Why do we need the chromatography as upfront of the mass spectrometer? Is because imagine that in the cell extract or in a tumor tissue, we have hundreds of metabolites. We could put them into the mass spec and analyze them, but the complexity will be overwhelming. So what people came up with, a very clever idea, was to separate the metabolites based on their physicochemical properties. And this is when the liquid chromatography is very useful. Liquid chromatography or gas chromatography, this is another approach, separate small molecules into different what we call retention time. So you pass this metabolite into a chromatography, and then metabolites will be eluted at different time depending on their physicochemical properties. I will give you an example in a second. Then each of these metabolite will be ionized and will be injected into the mass spectrometer. Again, the mass spectrometers nowadays are of many types. You have the time-of-flight mass spectrometer, you have triple quadruple, you have Orbitrap machinery. And I will give you an example about the Orbitrap, just to understand probably one of the most common mass spectrometer for metabolomics. The mass spectrometer will analyze each of the molecules that have been ionized from the chromatography, and will give what is called the M/Z, or its molecular weight. So imagine that now from each of the compound that we have from the chromatography, we can understand their molecular weight. And given the fact that nowadays the mass spectrometer are called high accuracy mass spectrometer, high resolution, we can infer the nature of the chemical based on their accurate mass. So based on the mass, we will know what metabolites are present in this extract, and then we can build our metabolomic analysis. Okay, so this is in a nutshell the pipeline. I told you that I wanted to give you a couple of examples of chromatography and of mass spectrometry. So with regards to chromatography, this is one example. There are different types of chromatography, depending also on the chemicals that you want to separate. But one of the most common and widespread is called helic chromatography. Here, the chromatography column is composed of silica beads that are this green thing here, that are then enriched with zwitterionic compounds. You can see, I hope you see my mouse, but this is the molecule that is bound to the silica beads. This provides hydrophilic surrounding for the molecules to get in. And then depending on the physical-chemical properties of the molecule, the more hydrophilic they are, the more they are retained in the column. And then we use the eluents that are hydrophobic and with a different degree of hydrophobicity to allow the chromatography to occur. Again, this is very general. I'm just telling you about this helic chromatography because you might have heard about helic or you might have read this in the method sections of some metabolomics paper. Just to give you an idea, apart from this helic chromatography, there are also reverse phase chromatography where the stationary phase, this is how it's called, the column is actually very hydrophobic, and this will retain more hydrophobic compounds such as lipids. Now, with regards to the mass spectrometer, again, I mentioned that there are many different types and different brands. The one that I want to mention is the Orbitrap, because again, you might have heard about the Orbitrap a few times. It's very popular because it allows this high resolution, but also high accurate mass determination. And this is how the Orbitrap is structured. So I told you that the ions coming from the chromatography are ionized. Ionized meaning that they go from a liquid phase to a gas phase, and they are ionized via what is called an electrospray source. So there is a high electricity and vacuum that allows the ions simply to fly. Imagine that now these ions in the liquid phase can fly into the machine, and then through various mechanisms and electromagnetic field, the ions are accelerated. They can get into this quadrupole where the ions can be filtered depending on their molecular weight range. And then they go through what is called a C-trap. I don't want to go into details. They go into the Orbitrap. Imagine now the ions will go from this nozzle here. They will fly and they will be accelerated in the mass quadrupole, and then they enter in this Orbitrap. In the Orbitrap, and now I will show you, the ions will start making exactly an orbit, and depending on its size, the orbit will be different. And the machine can infer the size exactly based on the orbit that it will make. And it does so by determining the intensity of the ion inside this the Orbitrap, and through what is called a Fourier transformation, it will give the m over z. That is the molecular weight. So you can see, just to give an idea, what is the Orbitrap, the size of the Orbitrap, eventually where all the ions are ionized and eventually they make this path, is not much bigger than a €1 coin. Just to keep in mind that this machine can be up to 1 million. So it's a very sophisticated piece of machinery, even though the core of this machine is very, very small. So now I give you this very, very broad overview of what metabolomics is. I gave you an example of a machine and of what is called liquid chromatography coupled to mass spectrometry. And I wanted to give you an idea of the applications. What can we do with these machines? And I will give these examples also using some of our science. So I hope it will make more sense. So I have divided the application of the metabolomics into 4. The first one is targeted analysis, and I will explain the other later. So targeted analysis means that with this machine, we can measure specific molecules, specific metabolites. And I will tell you a little more in details what we do, just to make sure that you understand why we look for specific metabolites. So as Roberta said, our lab is very much interested in understanding how mitochondrial dysfunction can cause cellular transformation, that is tumorigenesis. And we are interested in this field because a few years ago, now 20 years ago, it was demonstrated that mutations of mitochondrial enzyme of the Krebs cycle, it's a biochemical pathway inside the mitochondria, can drive hereditary and sporadic forms of cancer. And we are in particular interested in fumarate hydratase. And in a nutshell, fumarate hydratase was found to be mutated in a disease called hereditary leiomyomatosis and renal cell cancer. I will tell you about the disease in a second. Just to familiarize you with fumarate hydratase, I will tell you that this enzyme converts fumarate to malate within the Krebs cycle. This is all that matters for today. In 2002, it was demonstrated that mutations in fumarate hydratase can drive, as I mentioned, hereditary leiomyomatosis and renal cell carcinoma. You might have heard about this disease. It's called HLRCC. And without going into too many details, fumarate hydratase mutation will give rise to a very aggressive form of renal cancer called fumarate hydratase deficient renal cell carcinoma, along with very painful tumors of the skin and uterus. So now, why metabolomics is important? One of the key questions that we wanted to address already maybe more than 15 years ago was to what extent the loss of fumarate hydratase would change cellular metabolism, because until then it was believed that no cells could survive the loss of fumarate hydratase because it would be lethal. So we had to understand first whether the loss of fumarate hydratase would change the abundance of its substrate, that is fumarate. And to do that, we had to embark in what is called a targeted metabolite analysis. So here we used this machine, the Orbitrap, even though we could have used many other instruments that it's not really relevant, but we used the Orbitrap to measure fumarate. So here we perform a very targeted analysis. We needed to perform not only a relative quantification of fumarate in the wild type versus the fumarate hydratase knockout cells, but we wanted to know intracellular concentration of this metabolite. And to do that, we needed to perform, as you might remember from analytical chemistry or biochemistry or just chemistry, we needed to perform a standard curve. So we established a standard curve and we were able to determine the amount of fumarate that accumulates in the fumarate hydratase deficient cells. Again, fumarate accumulates because we don't have fumarate hydratase, and we found that fumarate accumulates to high millimolar levels in the cell, and we did this both in mouse and human cells. So metabolite analysis in a targeted manner allowed us to determine the intracellular concentration of this metabolite. I just want to point out a couple of things. When we say intracellular concentration, we actually mean the amount of this metabolite in the cell, not the amount of metabolite in the cell extract. So we have to infer, and to be able to infer the concentration of metabolite in the cells, and to do so, we need to know the cell volume. So when you embark in such a targeted analysis, if you want to determine the absolute concentration, just make sure that you calculate the cellular volume. And the cellular volume can be calculated by different mechanisms. It can be different instruments, sorry. It can be or inferred by simply looking at the cell shape and just inferring that is a sphere, even though this is not 100% accurate. So please remember to measure the cell volume if you want to measure the intracellular absolute concentration of a metabolite. Why is this relevant? Because very often when doing metabolomics, we want to know whether the intracellular concentration of our metabolites is sufficient, for instance, to block an enzyme for competition with a substrate or due to product inhibition. And in order to do that, we need to understand its absolute concentration and maybe the Km of the enzyme that we are studying. Okay, so this is the first point. You can also determine the simple relative concentration of a metabolite, but in that case, you have to have your relative comparison in order to make any inference. Now, the second point that is becoming much more fashionable is what is called semi-targeted metabolomics. And here, what I mean is the ability to analyze hundreds of metabolites from the same sample. This is really what metabolomics brought to the field of metabolism, the ability to analyze all these metabolites at the same time. And we can do this by analyzing the intracellular metabolites or the metabolites that are present in the media that are released by the cells. And I want again to give you a few examples of this. So here again, keep in mind that I'm always showing the example of the fumarate hydratase knockout versus the wild type counterpart. Here, this is how we present this ratio. This is a volcano plot. You might be familiar with this. This is something that we performed very recently, and you can see the volcano plot shows metabolites that are or accumulated in the knockout cells versus metabolites that are depleted in the knockout cells compared to the wild type. I want to point out, for instance, fumarate here. Fumarate is highly accumulated in the cells, but what you see surprisingly, is that along with fumarate, there are many other metabolites that are accumulated, argininosaxonate, metabolites that we didn't know existed, succinic glutathione and succinylcysteine. So I wanted to give you what we learned thanks to this semi-targeted metabolomics. We were actually able to pinpoint the source of these metabolites. And again, I don't want to go too much in details, but this is a schematic of what happens in our fumarate hydratase deficient cells. And we were able to show when the fumarate is accumulated to the high levels that I show, it changes a lot intracellular metabolites. A fumarate, for instance, the accumulation of fumarate can drive a change in the equilibrium of some reaction that would normally produce fumarate. For instance, within the urea cycle, there is a reaction that is called argininosaxonate lyase that normally produces fumarate from argininosaxonate and it releases arginine. However, when fumarate is accumulated to the levels that I show you, then the cells devise this very interesting mechanism whereby they take up arginine and they use the reverse of argininosaxonate lyase to produce argininosaxonate. And you can see that by mass spec, we were able, this is now a targeted analysis, we were able to show a significant accumulation of argininosaxonate. And the same thing happened, for instance, for other metabolites. I show you, for instance, metabolite called adenylosaxonate, and we can measure adenylosaxonate accumulated due again to the fact that fumarate leads to the reversal of another enzyme called adenylosaxonate lyase and leads to the production of adenylosaxonate. And finally, another set of metabolites that we found to be significantly accumulated. This required more investigation because these metabolites were not annotated in our semi-targeted analysis. But these are metabolites that come from a very peculiar reaction between fumarate, which is an alpha-beta unsaturated dicarboxylic acid, and cysteine residues of free thiols of cysteine and of glutathione. And we were able to show again that thanks to this analysis, we found that when fumarate is accumulated, due to this high accumulation, cysteine and glutathione are also bound to fumarate. And we were able to detect this in our semi-targeted analysis. Finally, the other point I wanted to make is that when you do this semi-targeted analysis, not only you can infer information about intracellular metabolites, but also the metabolites that these cells secrete. And this, of course, is very important if you want to understand whether there are changes in what we will call metabolic fluxes. And here in this work, we were able to show, for instance, that the fumarate hydratase deficient cells take up a very different amount of nutrients. And this was very informative because then it linked this to, for instance, protein synthesis and integrated stress response. But I don't want to go into detail. But we were also able to show that fumarate, for instance, is very actively secreted in the media of the fumarate hydratase deficient cells, and also argininosaccharide, I mentioned, can also be released in the media. So by analyzing the media of your conditions, you understand better whether some metabolites are differentially taken up or released. And of course, I strongly recommend this analysis to have a better overview of what we call metabolic fluxes. A couple of notes. When you perform steady-state metabolomics, that is the analysis of these metabolites in the cells intracellularly, your input should be always normalized. We don't want to, as for any other analysis for Western blot of qPCR metabolomics, data have to be normalized for the input, and there are many ways to normalize for input, and we can discuss this later. But one way is to start with the same amount of cells extracted with the same amount of extraction buffer. So you start with an equal amount of total metabolites being analyzed. Another very important point that I want to make that I often hear, maybe at presentation or during revision of the papers that Roberta mentioned, is that when we do this steady-state metabolomic analysis and they are not absolute quantifications, we cannot compare different metabolites with each other. For instance, if we, in the graph, you see different area of the peak of fumarate versus succinate, we cannot infer that fumarate is differently concentrated compared to succinate, because there are important parameters of each metabolite, such as, for instance, ionization efficiency, that affect the actual performance of the ion in its process through identification in the mass spectrometer. So we cannot establish whether a metabolite is more concentrated than another using these approaches. If we want to make that conclusion, we need to determine the absolute concentration of the metabolite. What we can do, though, is we can compare the same metabolite, let's say fumarate, in two different conditions, in your wild type and knockout, because in this case, the metabolite is the same, and it's in the same cellular environment, is in two different subtypes, but the environment, chemical environment is the same. Therefore, the performance of the peak can be confirmed. And we can therefore say that fumarate is higher in a condition than in the other. Second point is that for consumption release experiment, where you measure the concentration of metabolites in the media, of course, it's very important to determine how many cells did you use during this experiment, because of course, if you have conditions where the cell growth is very different between two conditions, of course, the amount of metabolites that is released by one cell will be intrinsically different for the other and will not tell us much about the intrinsic release of a metabolite. I hope this makes sense. But again, I'm happy to cover all these points later on. Okay, yes, I mentioned all this. The other type of analysis that can be done with this metabolomic pipeline is what is called the untargeted analysis. You might have heard about this more often, but there is a very, very important difference between targeted and untargeted metabolomics. In the targeted or semi-targeted metabolomics, we start with a number of metabolites that we know and that for which we know chemical nature, maybe the retention time in the chromatography, and therefore we have a list of metabolites that eventually we will extract from the analysis. With the untargeted metabolomics, we will actually collect all the results that come from the machine, and then we will do a very complex alignment of the data, of the peaks, in order to attribute the chemical nature of all these metabolites that we will call feature. So it's completely blind analysis. We will take every molecule that comes out of the chromatography, and we will analyze. We will determine the ratio between the two conditions, and eventually we using a specific software, we need to infer their name. However, this is very prone to error. This is very difficult and very intense. Here, I just want to give you an example of a proper untargeted metabolomic analysis that we did in the lab very recently, maybe last week, just to show you the difference. So this now is the same cell type that I showed you before. So the fumarate knockout versus the wild type. And you can see now many more metabolites. We have now 500 metabolites that we curated. And initially, you can get from the analysis more than 8,000 features. But then we need to filter a lot of noise. And then we need to also figure out the name of these metabolites in the best accurate way possible. But you can see that, for instance, the metabolites that we described earlier, such as fumarate and argininosaxonate and saxinocysteine, are still there, but there are many other metabolites that now appear and for which we actually don't know much. And this is one of the negative things about untargeted metabolomics, is that you will find this metabolite with a very long name, and eventually you might not know what it does. And this is very often the case with this type of analysis. So if you want to do untargeted metabolomics, you need to be aware of the fact that the analysis is much more tricky, is much more prone to misannotation, and you need to have a much more robust biostatistical pipeline there, along with the analytical pipeline. And you need to perform the appropriate, there's a lot of, let's say, normalization that has to go into the pipeline. What I call here MS2, that is MSMS, meaning that you need to analyze also the fragment of each metabolite in order to be able to impute the correct name for the metabolite. So this is much more intensive, but on the other side, it can give you some true new metabolites that might have a role in your biological condition. So finally, something that you might also have heard, I wanted to talk about metabolic tracing, why this is important. So when we analyze metabolomics or in the cell or in a muscle, we call it steady state metabolomics. And imagine it's a snapshot. So imagine that metabolism is just traffic. We take a snapshot of the traffic at a given time as indicated here. However, we don't know whether that photo shows a stacked traffic or whether the traffic is proceeding very fast. So for instance, we might have a condition where a metabolite is very high, but it doesn't mean necessarily that the metabolic pathway that produces it or that degrades it is blocked and there is a congestion in that specific metabolic node. It might simply be that there is a lot going on in this metabolic pathway, and we do not know whether this is the case by simple steady state metabolomics. So people devised another approach, which is called isotope tracing. How does it work? With isotope tracing, we give, for instance, a substrate that, like glucose or glutamine or any other metabolite that will be converted into your metabolite of interest. And this heavy isotope can be distinguished for the natural one via high resolution mass spectrometry. And eventually we can perform time courses or single time points, but we can establish to what extent this specific metabolite that we give is converted into another one very accurately. So the pipeline for carbon labeling experiments, and by the way, I very often refer to 13C carbon labeling, but this can be done with any other natural occurring heavy isotope. 13C carbon labeling is very widespread, is very common. Of course, this is not radioactive. It's a heavy isotope, but it's non-radioactive, so you can use it safely. We have performed experiment with heavy glucose, for instance, in humans. It's absolutely no problem. But it requires a lot of computational analysis downstream. So you will need to correct for natural occurring isotopes. This is something that I will mention more in detail later. And you can perform two things. You can do one time point steady state 13C labeling, and for which you will be able to infer just the activity of a pathway, or you can perform dynamic labeling, meaning that you will perform multiple time points, and with this you can determine metabolic flux. Just to go a little bit deeper into what I mean by metabolic state, steady state and dynamic labeling. So imagine that you give your cells 13C carbon labeled glucose to investigate glycolysis and the TCA cycle. You need to give this glucose for a long time enough for until all the glucose is being utilized and all the metabolites in the pathway that you want to investigate have reached the steady state. So they have reached their maximum labeling capacity. Or you can do something different, that is to give your carbon labeled glucose, and then you perform time points. And at this point, you will see enrichment of different metabolites that will change over time. And this is exactly what will be used to determine the metabolic flux. And imagine just here that the more you leave your 13C carbon labeled compounds over time, then the closer you are to reaching the steady state. So the steady state labeling that's here, it's exactly the one here, and the dynamic labeling will be performed before they reach the steady state. So with the labeling analysis, what the mass spectrometer will do, it will analyze what we call the isotopologues, meaning how many carbons or how many isotopes, heavy isotopes, are enriched in your molecule of interest. And this will generate what is called a mass distribution vector. I know this is probably very technical, but it will become very clear in a second where I show the picture. The bottom line is for your molecule of interest, you need to establish the heavy carbon enrichment for every carbon, and eventually you can present this as a simple ion count. So each isotopologue enriched from the carbons, a heavy carbon, will give you an intensity. Or you can calculate this as a fraction of the total, meaning that eventually, let's say that this is pyruvate, okay, which is a 3-carbon molecule. The 3-carbon molecule pyruvate can have only 1 carbon, 2 carbons, or 3 carbons coming from the heavy glucose. And the mass spectrometer will give you the intensity of each of these isotopologue. You can calculate the total of this, which will give what is called the total pool of the pyruvate, and then you can calculate the fraction, meaning how much of the pyruvate is unlabeled, how much of the pyruvate is labeled N+1, how much is labeled N+2 and N+3, and you calculate this mass distribution vector of the fraction. Now, I know it sounds maybe complex, or maybe I wasn't able to explain it very clearly. These two options are valid. But here, I hope I give you something that will clarify this. So I told you that we work on the TCA cycle, the Krebs cycle, and you may know that one of the main substrates for the TCA cycle is glucose. So glucose is converted by glycolysis, and then the carbons from pyruvate, which is the endpoint of glycolysis, are then further oxidized within the Krebs cycle. Now, if, as glucose is a 6-carbon molecule, if we give fully labeled glucose, meaning that each of the carbons indicated here as a dot are 13C, they will be converted into a fully labeled pyruvate. So each carbon of pyruvate will be labeled N+3, and then pyruvate will be oxidized via pyruvate dehydrogenase into acetyl-CoA. Now, pyruvate and derive acetyl-CoA, which is now labeled, as you can see, with 2 heavy carbons, will be condensed in the first committed step of the Krebs cycle into citrate via citrate synthase, but it will use the 4 labeled carbons from oxaloacetate. So now a completely unlabeled oxaloacetate will be condensed with the label acetyl-CoA, giving rise to what we call an N+2 citrate. So citrate is a 6-carbon molecule, and only 2 carbons will be labeled from glucose, giving rise to a shift in the molecular weight of citrate of 2 daltons. Now, if we proceed in the Krebs cycle, 2 unlabeled carbons will be removed through the series of reactions, and fumarate, which is now a 4-carbon molecule, will be labeled again N+2. Only 2 carbons will be labeled from glucose, and this will shift the molecular weight of fumarate of 2 daltons. As you can see, this is what I call the mass isotopologue distribution. With the mass spectrometer, we can determine the percentage of each of these isotopologues of fumarate in the 2 different conditions. What you will see, I hope it's clear, is that, for instance, the N+4, N+2 fumarate, which is the one that is labeled from glucose, is very reduced in the fumaratidase deficient cells. So what this means is that the contribution of glucose into fumarate is reduced in the fumaratidase deficient cells. So this suggests that something else might be contributing to the fumarate pool. And we found that one important contributor of fumarate is actually glutamine. Sorry, 1 second, I've seen something up here. Okay. So glutamine is a very important substrate for the Krebs cycle, and you have heard this probably many times. Glutamine is involved in this reaction called glutaminolysis. How it works is that the glutamine is converted directly via glutamate into alpha-ketoglutarate, and glutamine is a 5-carbon molecule. And when when we give fully labeled 13C carbon labeled glutamine, it will give rise to 5 labeled alpha-ketoglutarate, and this will be converted into a fully labeled 4 carbon molecules coming from glutamine. So now, if we look at fumarate N+4, which is the one that is produced by glutamine, we will see that indeed, quite intriguingly, there is a significant increase in the carbon labeling from glutamine compared to what we showed before. So this suggests that in the fumaratidase deficient cells, glutamine is the main substrate for fumarate production, whereas glucose, it's reduced. Now, putting things into perspective here, we have the carbon labeling from glucose and we have the carbon labeling from glutamine. Presenting the isotopologue distribution enable us to see that glutamine, which gives rise to 4 carbon label, is the main contributor of fumarate pool, whereas glucose, which contributes to an N+2 label fumarate, contributes very little to the fumarate pool. And this allowed us to conclude that when we lose fumaratidase, glutamine is the main contributor of the TCA cycle and the fumarate. I hope this makes sense. But this approach was actually very useful in the past to identify several behaviors of the Krebs cycle. For instance, it was thanks to this labeling of glucose and glutamine that we were able to, not we, but we and others, to show that, for instance, glutamine can undergo what is called reductive carboxylation, whereby the glutamine, instead of being fully oxidized, as I showed you before, within the Krebs cycle, can also be used to produce citrate and eventually for citrate to be exported. But we have also other behaviors where glutamine can be utilized, for instance, for generating even lactate, and now we know even to contributing to glucose. So why it is important to perform a labeling experiment? It's because here I'm plotting side by side the intracellular quantification of fumarate that I showed you at the beginning, and here I'm showing you the labeling from glutamine. So you can see, first of all, that the two type of experiment give a very different result. With the absolute quantification, we will only know how much fumarate is accumulated in the cell, or a metabolite is accumulated in the cell. With the glutamine labeling or any other type of labeling, you will know what substrate contribute to that specific metabolite. However, it is very important to present both pieces of information in a paper, because as you can see from this presentation, when we show only the percentage of labeling of glutamine into fumarate, we would never have guessed the fumarate was so highly accumulated. Just by looking at the percentage of labeling, we would say, okay, yeah, fumarate is doubly labeled from glutamine than in the wild type compared to the knockout. But it's only through showing the total pool and the quantification of the metabolite that we will have a better understanding how much the metabolite is accumulated. So to reiterate, when you want to present your labeling experiment, please always include the total pool size in order to have a better picture of what you're measuring. So all this information, all this guideline is included in a recent paper that Johannes Meiser and I written in ENBO, just to give a guideline of how data should be presented. All the caveats that I mentioned, for instance, the making sure to measure the cell volume for absolute quantification, making sure, for instance, to subtract the natural abundance of a metabolite from your labeling experiment, and so forth. The last point I want to make is about metabolic fluxes, because I hear very often people conflating metabolic tracing with metabolic flux analysis. So when you perform one time point metabolic tracing experiment, you shouldn't be talking about flux analysis. Flux analysis should be performed only when you determine the actual enrichment of a metabolite from its carbon source by looking at it over time before it reaches saturation. So instead of measuring only the end point, you should be measuring different time points before the metabolites reaching the isotopic steady state. And why it is important to do this? Because only via this measurement we will be able to assess the actual metabolic flux within a reaction. So to perform a proper metabolic flux analysis, you will have to take into account the uptake of a metabolite, the secretion of the product to the reaction, and eventually calculate the rate of conversion of this metabolite. And this can be done, of course, via this very time-resolved metabolic analysis with the carbon labeling, but eventually you will need some computational analysis. Here I put in just a guideline to show that together with the 13C metabolic analysis and this metabolic modeling, you can come up with accurate determination of fluxes. Okay, and by fluxes I mean the actual conversion in micromole per minute of a metabolite into another one. And for this, again, you need to establish this metabolic modeling that requires a completely different discussion at this stage. Oh, okay. With this, I will conclude. I just want to thank the people in the lab who have done this experiment that I show you, some of the funders and the collaborators, even though I didn't talk too much about our science. But I think I would reserve the remaining time for questions because probably there are many things that you want me to clarify.
Thank you, Christian,
Okay, I
for
will leave.
this clear and educational and important piece of information that you provide us.
Thank you.
So we have some questions in the chat. So Fabio Penna, thanks for the very informative lecture. Can you suggest public repositories to search for already existing metabolomics datasets?
Oh, yes,
As
excellent question.
far as I'm concerned, open science in metabolomics is still a bit insufficient or lacks a consensus on public data availability. It's an
Yes,
important point.
yeah, Fabio, thanks. This is a very important point. Unfortunately, there is no consensus. I agree with you. At the moment, each dataset that someone produces should be uploaded into either one database, which is called Metabolites, which is curated by EBI, so the European Bioinformatics Institute, and you can find all raw data generated by many, by a lot of experiments in that database, or another one that is instead from California. It is called Metabolomic Workbench, and again, you can find a lot of data sets in these databases. There is a caveat there, of course. Very often people just put raw data, so you will have to be able to process this data, and this requires still some work on your side. There is still no requirement, official requirement for metabolomics data deposition in any of these. Some journals now are making it compulsory to deposit the dataset. So we are not there yet as we are for genomics or proteomics, but things are getting there.
Related to this issue of the raw data, so when you
Yes.
send your samples to a facility, you have back the raw data. So do you have any suggestion on public available program, software, resources that one that is not able to use R or other kind of program, and user-friendly platform that can be used to elaborate the date and to have a graphical output? Is it dangerous? I mean,
Yeah.
what is your opinion in that? Because
Yes.
in the vast majority of the case, you have back the raw data. And if you want to have the data analyzed, you have to pay a lot of money. And so
Yeah.
people try to use by
Yeah.
ourselves.
Yeah.
So what's
But
your opinion?
Roberta, first of all, it depends what you mean by raw data, because I doubt that the people, the facility will give the actual mass spec data, which are raw, because then you will have to use proprietary software to go from the raw data to the peak to the peak area, okay, the metabolite intensity. So by raw data, do you mean Excel spreadsheet or?
Yes, exactly.
Okay,
Exactly.
okay, okay. So that's a different story. So once you get the data from from the facility with your intensity and your metabolite, then you can use some. There are software out there. For instance, one that we find increasingly getting better, it's MetaboAnalyst.
Mm.
You might have used this.
Yes.
MetaboAnalyst will do quite a lot nowadays. It will, first of all, you can, if you know R, you can use all the scripts there, but it will give you metabolite data presentation. So some enrichment analysis, so metabolic pathway analysis. So this is probably the one that I would recommend
Okay.
for if you're not heavy user. Then if you are a heavy user and then you want to delve deeper, then things become more complicated and you need to do a lot of manual searches, unfortunately.
Okay.
Um, we
Thank you.
also have, again, there are some initiatives. I didn't want to promote this here, but for instance, our lab has created a package that you can, if you are an eye user, you can download. It's called Metaproviz. It's for metabolite visualization and analysis. I think I have it. Yeah, it's this one. So if you want, again, I'm happy to share the slides. Christina Schmidt, she was a PhD student in lab, worked quite extensively on being able to go exactly from your data Excel into differential metabolite analysis and also visualization in a way that can be more tailored because MetaboAnalyst doesn't allow that. So for instance, with MetaProvis, you can get principal component analysis, you can get heat maps based on how you annotate your metabolite. You can have specific visualization of individual metabolic pathways. For instance, here you can ask the script to look only for the TCA cycle and will give you heat maps. And eventually it can give you volcano plots or other type of visualization like this bar graphs. Yes, I think
Thank you.
that is all. So if you want, you can download it. We are working on getting this published, but it's already available.
Thank you. So Daniele Renzoni, how a non-annotated metabolite can be identified?
Oh, yes, this is extremely, extremely complex. And you need, of course, from a mass spec point of view, you need to uh, rely on MSMS, so fragmentation of the peak, in order to be able at least to establish, based on the fragments, what molecule it could be. Nowadays, there are a lot of tools to infer the chemical structure based on this MSMS and based on MS. Depending on the software you use for the mass spec, you can do this. But eventually, if you really want to uh, figure out what is the chemical molecule that you have in front of you, you need to perform NMR, so orthogonal analysis. So you need to use other machine. For instance, the magnetic resonance is definitely extremely useful in these conditions. And eventually, final confirmation, you need to create this compound and then you compare with your unannotated peak. So if you really want to get there, it requires a lot of energy.
Another question from Nezibe Peker. What do you think about normalization methods in tissue metabolomics? Is it possible to compare metabolite levels among different tissues using absolute quantification method? If so, do you think that is still reliable given that different tissues have different cellularity and density? Another
This
crucial point.
excellent. Excellent point. So what we do at the moment with regards to tissue, we normalize for um, amount of tissue that we extract. So let's say we take 2 milligram of tissue for each tissue that we want to analyze. Of course, this is very much prone to problem, especially because different tissue might have different interstitial fluid, again, different cellularity, and also different tissue have also different components, meaning different cell types. So some tissue might be more infiltrated with T cells, so you have extreme heterogeneity. But at the moment, the best we can do is to analyze the same amount of tissue to start with. But at the moment, we can only provide absolute quantification, meaning how much metabolite per milligram of protein of that tissue, but we cannot infer the concentration per cell unless we determine the cellularity. This is something that's so tricky to do that we haven't been able to do it. And I haven't seen this published yet anywhere. So bottom line, just to address your point, we can still compare metabolite in different tissue by just comparing the either relative quantification or the absolute quantification, but determined per milligram of tissue.
And this is what you are referring when you say to calculate the cellular volume, or
Yes, because from a tissue, you know, if it is a cell culture, we can infer cellular volume based on how many cells we have and the average volume of the cell. But for a tissue, it is very difficult to extract how many cells you have in those 2 milligrams, and also even more difficult to extract the average cellular volume. So, yeah.
Yeah. So personally, I have many other questions, but the time is over because it's 6:06. So Christian, thank you again for this fantastic overview and for preparing this for us, for this experiment that we did today together.
Yes, it's an experiment.
Thank you again. It was very useful and clear.
Thank you very much to all of you for bearing with me, and thanks a lot for the question. They were absolutely spot on. Thanks.
Yeah. Okay.
Okay.
Thank you all. So see you in April now for the trainee talk, and have a